name is Xintong. I'm assistant professor at the University of Texas at Dallas. I'm in the same department as Michael, soon to be my ex-colleague, Dr. Denisko, and also Dr. Larry Reiser, who work in the UTI field. My lab primarily works on this group of peptides called antimicrobial peptides. And we are actually interested in the functions of these peptides across a number of epithelial barriers. So everywhere where our body interact with the microbial world, we have the epithelial tissues expressing these peptides actually. This includes our skin, which of course covers our whole body, our airways, the gastrointestinal tracts, and our urogenital tracts. Usually when we're healthy and at a steady state, we do not have a lot of these AMPs expressed. So we have a few, they're not supercharged. They kind of keep the microbiota in balance. However, if an infection comes in or in certain inflammatory diseases through immune dysregulation, our epithelial cells get inflamed and they start to pump out these antimicrobial peptides in pretty large quantities. They are called antimicrobial peptides because they were first discovered for their antimicrobial activity. So if you have a dish of bacteria, you drop some of these peptides on it, the bacteria is going to die, both gram-positive and gram-negative. However, over the years, our lab and other labs have found that these peptides also perform signaling functions. And we are primarily focused on the innate immune system. And we are focused on a few types of innate immune cells that, in our opinion, includes the sensory neurons. I'll talk a little bit about it in the end. And these granulocytes called neutrophils, mast cells, and basophils. And we found that each of these cell types express a cell type specific G protein coupled receptor that allow them to respond to these antimicrobial peptides as a signal. And these GPCRs all belong to a large GPCR family called mass-related G protein coupled receptors. It's a mouthful, and it is my postdoc advisor Xintong's fault that it's a mouthful because he named them. We usually just call them MERGs. And in my lab, people start to use an abbreviation. They just call the mast cell version of it B2 instead of MRGPR B2. When Xintong first found these receptors, he cloned them out of the sensory nervous system. So he was a postdoc in David Anderson's lab at Caltech, and they were studying gene expression using the fancy technology back in the 90s called the microarray that none of us use routinely anymore. But they had a mouse that doesn't have any nociceptive neurons. And he performed an array study comparing wild type and these mice that don't have nociceptive neurons. And that's how he first discovered this gene family. And he spent years characterizing the neuronal members of these GPCRs and made a lot of contribution to the sensory neurobiology field. He is credited as the discoverer of the neurons that make us itchy. And over the years, people in the field start to realize that in addition to the nervous system, you actually can find other members of this family in the innate immune system. The most famous and well-studied and heavily kind of pharmacologicalized member is this red one called MRGPRX2. It is expressed specifically in connective tissue mast cells. And I'm actually going to talk a lot more about these cell types. In the skin, it is the major source of histamine, and that is what gives us itchy and inflammation during allergy. I joined the lab a while ago, and I was a postdoc fellow, and during my postdoc studies, I actually characterized another two new members of the family. One is called MRGPR-A6. This member is expressed in another type of granulocyte called basophils, which are kind of the circulating counterpart of mast cells, also source of histamine. And I also study this other member called MRGPR-A2 that is specific for neutrophils, which are, of course, our heavy arsenal for antibacterial immunity. And on the antimicrobial peptide side, the major families of AMPs in the mammalian systems are called defensins and cathelicidins. And these peptides are all pretty famously cationic, meaning that they bear a number of positive charges at neutral pH. And we actually found that the ligand receptor interactions between these peptides and the GPCRs are based on charge as well. One of the receptors we study, this mast cell receptor, MRGPRX2, has actually been solved by cryo-EM. And the structural biologists found that this receptor has a shallow but negatively charged binding pocket that allows it to respond to a number of cationic ligands, including these antimicrobial peptides. And another member called MRGPRX4 is kind of the negative control here, has a positively charged binding pocket, so it actually does not respond to any of these peptides. And we found that the more charges the AMP bears, beta defensin, alpha defensin, or cathelicidin, the more likely it is going to activate these receptors. And we have also studied the mouse homologs of both the peptides and the receptors, and we find kind of the same story. And our observation is that for the tissue resident cell types, there are sensory neurons, there are mast cells, and they are sitting in the tissue at all times and performing a sentinel function to detect anything mildly wrong. These cells have receptors that respond to the peptides at lower EC50, and they're more sensitive. Whereas the neutrophils, which are the heavy hitters, and we don't want them activated at all times, they cause tissue damage, have a higher EC50 and respond actually only to the most cationic peptide among all the ones we tried. Xintong's lab over the years, myself included, have built a pretty huge collection of genetic tools on these receptor families. He has single, double, cluster knockouts. These, of course, allow us to perform necessity studies to look at the loss of functions of each of these receptors in their corresponding cell types and context. And because these receptors are fantastically cell type specific, we also took advantage of this specificity and built a number of CRE lines that allow us to selectively label, visualize, manipulate, ablate, activate, inactivate these cell types. And he has also built a number of humanized mouse lines to replace the mouse receptor with a human version. This allows us to test out the pharmacological kinetics of these receptors using a mouse model. And on the ligand side, defensins and cathelicidin, as I said, are the major families. Cathelicidin is a single gene, and the knockout was made by Dr. Victor Nizet and Richard Gallo at UCSD in the 1990s. The defensins have been a lot of trouble to study genetically because they actually are highly genetically redundant. In the mouse, there are over 50 members clustered onto the ACE chromosome and spans over 3 million base pairs. But I entered the field, I was really naive. And Xintong loves making cluster knockouts. So he's like, oh, a gene cluster, let's make a cluster knockout. So it took me 3 years to finish this. We inserted 2 LOXP sites on the 2 ends of the gene cluster and homozygous the flox allele, and for our initial study in the skin, crossed it with a keratin 14 CRE to generate this huge 3 million base pair cluster deletion. And the day I run this gel was a very good day in my academic career, because we did see that this CRE-LOX system does give a robust deletion of this entire gene cluster from the skin. You can see that if we sequence this deletion band, you see the 2 LOXP sites recombined into 1, and they're flanked by the 2 ends of the cluster. And we've validated this on the RNA level and also the peptide level for one of the highly expressed members. And we see that the knockout is very clean. In the skin, we use Staph aureus as our primary model pathogen. We use the luminescent version of the bacteria. So you inject the bacteria tag with a firefly luciferase, which allows you to monitor the level of infection over time without having to sacrifice the mice and take CFU every day. And our collaborator has validated that the luminescence and CFU is in a very, very tight linear relationship. Pretty obviously, and not surprisingly, if you delete all these defensive peptides, the mouse does have an antibacterial immune defect. They allow the bacteria to spread to a much larger area, have higher bacterial burdens throughout the course of infection, and the infection will cause a much larger necrotic lesion. And the kind of surprise we found there is that if we just knock out the neutrophil receptor, I don't know what my mouse is doing, I'm sorry, neutrophil receptor called MRGPRA2, we actually get almost the identical phenotype. You also get a much larger bacterial burden and large necrotic lesion, which is very interesting and really speaks to the importance of the signaling function of these peptides. We also look at the histology of these infections. In a skin Staph aureus infection, you see this very classical neutrophil abscess formed around the site of infection. And if you don't have either the defensins or the neutrophil receptor, you see that the neutrophil abscess is largely defective and the bacteria, as we've already seen on the IVIS, spread out to a much larger area. So without going into too much detail, we found that if you ablate the defensins or the MRGPRA2 receptor, you're jeopardizing this neutrophil pro-inflammatory signaling loop. Where activation of these neutrophils by these antimicrobial peptides will trigger the neutrophils themselves to release pro-inflammatory cytokines and chemokines to recruit more neutrophils and propagate these inflammatory response. And in the absence of this signal, we get this clear neutrophil abscess defect and also clear antibacterial defect. So this was my postdoc work and was published in 2022 when I was also on the job market. And out of I think 5 or 6 ideas that I pitched to Xintong, he was most excited about studying the same system in the urinary system. Because we know already, I've done a couple preliminary experiments to look at the urocilium, and I know that these bladder epithelial cells also express a number of defenses, different members, but there are some overlapping between the skin and urinary defenses. And we also know that the bladder has a somewhat analogous immune system with the same types of tissue resident and also recruited immune cells. And they also have sensory neurons, which is always my favorite cell type to study. However, these two host microbe interfaces differ in very important ways. The skin is an air interface, and the bladder, of course, is a liquid interface. And if you look at the microbiota, they're completely different. And the immune cells, although you are looking at the same type of cells, they assume tissue-specific phenotypes, which are very, very important for the functions of these cells and the overall immune system. So when I interviewed at UTD, I pitched this idea to Nicole and Larry, and I think I convinced them to hire me. And I started at UTD in 2023, and I wrote a R01 called a CATS R01, which is a kind of peculiar NIH funding mechanism that basically forces the young investigators to switch field and get out of your comfort zone and dive into a new field, but contribute your expertise to the new field. And I was lucky to have 3 co-investigators, Maria, Nicole, and my colleague Ben Kober in the Department of Neuroscience to support this grant. And I proposed to study the antimicrobial peptide signaling system in the bladder. In aim 1, we propose to make a bladder-specific defensive knockout because unfortunately the K14 Cre actually doesn't work in the bladder, so we have to use a different Cre line. In aim 2, following kind of a similar logic, I want to study the receptors of these antimicrobial peptides on a variety of immune cells, including the neutrophils that we saw was very important in the skin, mast cells, basophils, and also potentially dendritic cells, macrophages, and also adaptive immune cells. In aims 3, we want to look at the sensory neuron and their contribution to UTI-related pain and also neuroimmune interaction. So this grant was very fortunately funded, and aim 1 is currently ongoing. We have tried 2 Cre lines so far, UPK2 Cre and K5 Cre, where we see a partial deletion with UPK2 Cre because I think theoretically the defenses are not only coming from the intermediate and umbrella cells. K5 Cre unfortunately didn't give a deletion, and I was at the UTIGA meeting last week and I was talking to Dr. J.D. Spencer at Nationwide Children's Hospital, and he suggested I try Sonic Hedgehog Cre, so we're going to try that. Meanwhile, I have alluded to another important cationic antimicrobial peptide called cathelicidin, which is a single gene in the mouse genome and has a knockout mouse available since the 90s. And Dr. Hunstad's lab actually have already looked at the UTI phenotype in this mouse, and they use the same model that we do, where we in fact 7 to 10 weeks old C57 females with 10 to the 7 CFU of UTI 89. And to their surprise, they actually see that the mice that don't have this antimicrobial peptide actually do better than the wild type across a number of time points. And we have been able to recapitulate this phenotype in our own lab because we happen to have this mouse for our skin study. And this is very intriguing because and counterintuitive because you would guess that an antimicrobial peptide should be contributing to killing the bacteria in the bladder, right? Whereas you actually see it doing the opposite here. And we have been hypothesizing that the reason why we see this phenotype is actually because of the signaling function of cathelicidin. We have actually known for years that antimicrobial peptides, including cathelicidin, activate this mast cell receptor MRGPRX2 on the on specifically connective tissue mast cells. These cells are found in all organs of our body, and they are tissue resident, meaning that they sit actually pretty close to the barrier tissue, and they do not need to be recruited from blood when an inflammation is happening. And they're mostly well known for their functions in allergy. This receptor was first cloned from in the Japanese lab back in the 2000s, and Xintong found the mouse homolog called MRGPRB2, and we already know that it performs a number of very important functions in both allergy and barrier immunity. So we also know that the signaling function of cathelicidin strictly depend on this receptor. So if you assay mast cell degranulation in response to stimuli by cathelicidin, and you try this in a wild type cell versus a mutant cell, you'll see that the mutant cell does not respond to cathelicidin at all. And I would also like to mention that this project is a collaboration between me and my postdoc lab mate, Dr. Pandir and Green, who are at the University of Guelph and UTMB. So this is a beautiful image from Dr. Brian Kim and Hongzhen Hu's labs at Mount Sinai, where they use the light sheet microscopy to look at all the mast cells in the whole bladder. And they're not the most numerous cells, but we do see them distributed in kind of all layers of the bladder, including the lamina propria and some here and there in the urothelium. We use a reporter mouse called MRGPR-B2 TD tomato mouse that specifically label these B2 positive mast cells, and we see about the same thing. A small number of cells can be seen in the lamina propria and very, very rarely in the urothelium. And Warris has analyzed a single cell data set published single-cell data set, and we see the same thing. These MRGPRB2 positive mast cells are indeed seen in the bladder. And when we look at the CFU phenotype, we see the same as the cathelicidin knockout. The mice without this bladder receptor are doing better than the wild type. Meaning that in the wild type mice where this receptor is activated, it is actually doing something there that is not helping the host to clear this bacteria, right? We performed bulk RNA sequencing on these knockout animals, both infused with PBS as control and also infused with UTI-89, and we take the bladder 24 hours after infection. If we compare the genes that are expressed between the wild-type UTI bladders versus the MRGPRB2 knockout UTI bladders, we see a number of genes that are highly expressed in the wild type, but comparatively lower in the knockout. And these genes are mostly representing the immune pathways. They can be grouped into the leukocyte migration and chemotaxis pathways. And if you look at the gene set, there are all the interleukins and cytokines and chemokines. We also validated these using qPCR and also look at the infiltration of immune cells into the bladder using flow cytometry. And the flow result confirms that indeed we actually see a reduced number of immune cells infiltrating into the bladder following infection in these MRGPR-B2 mast cell receptor knockouts. And we see overall reduction in CD45 cells as well as neutrophils. So this kind of poses an interesting dilemma because we see the same thing happening in the skin and the bladder immune system. When the infection happens, the epithelial cells release the antimicrobial peptides. They activate this mast cell receptor in both the skin and the bladder. And mast cells degranulate and release factors to recruit the neutrophils to the bladder. And all of these are the same in the skin and the bladder immune systems. Whereas we know in the skin, this immune response is beneficial for the hosts to clear the bacteria. Whereas in the bladder, this is counteractive, right? And we were looking at this, and we think the major difference between these two systems lies within the epithelium itself. Because the bladder epithelial cells famously exfoliate after infection, which can potentially expose the deeper layers to more bacterial infection. And because it is a liquid interface, these cells are flushed away with the urine, whereas the same does not happen in the skin. So we look at bladder epithelium, and we see that pretty clearly the B2 knockout post-infection bladder epithelium is sicker compared to the wild type. Wild type is here. And our undergrad student Zara did a lot of quantification. She was completely blinded to the genotype or the treatment of these images. And we saw that indeed, these knockout mice have a sicker urothelium post-infection, which is also reflected in the RNA sequencing data set, where we see pretty much all the uroplakins gone from the wild-type mouse post-infection, whereas the B2 knockout still retain them. We are still working on this phenotype. We have been taking a 24-hour time point. So we are still resolving whether this is due to a reduced exfoliation or increased proliferation or potentially both. Another very interesting phenomena that Uris looked at is on the bladder cells receptivity to UPAC invasion. This is actually following a really, really good suggestion by Dr. Alice Shea at the Cold Spring Harbor meeting that Uris attended last year, where she came to his poster and basically pointed at it and then said, you should look at EGFR. In addition to EGFR, Uris also looked at the integrin genes, and we saw the same thing. For the wild type, after infection, also still 24 hours, we see this massive increase in EGFR and these integrin expression. This is particularly striking because if you compare this to the UPKs, you're actually seeing the reverse, right? And the B2 knockout, we see a much milder response. And in the case of the EGFR, we don't see any increase at all, despite them having a much thicker urocilium. And this leads us to the model where we have the B2 mast cells triggering some sort of gene expression changes through mechanisms that are still under investigation that induce the urothelial cells to overexpress these EGFR and integrin genes that allow the UPAC to invade more into the bladder epithelial cells. Consistently, we see that if we sample the urines CFU early on during the infection, we actually see more bacteria in the urine of the mutant animals. So that's all we know so far for this story, which is currently under review. I think the kind of bottom half of this diagram is pretty solid. During infection, we have cathelicidin, among other antimicrobial peptides released, and it activates this receptor on the mast cells, cause mast cell degranulation. And this, just like in any other tissue, recruits and activates other immune cells, including neutrophils. However, in the bladder specifically, this either directly through the release of mast cell mediators such as proteases, or indirectly through other immune cells such as neutrophils through its reactive oxygen species or other mediators, regulate the behavior of the urothelial cells, potentially triggering their exfoliation, proliferation, and gene expression that allows UPAC to invade the urothelial barrier and actually exacerbate the infection. There's still a lot of unknowns in this story. We really want to know the exact molecular mechanism of this mast cell urothelial crosstalk. We also want to know what is the happening to the UPAC bacteria because we actually see this really interesting phenotype where at a pretty early time point, 6 hours post-infection, in the B2 knockout bladders, we see a lot of filamentous bacteria in the bladder that we do not see in the wild type. So we're really interested in what this is doing. Another pretty cool phenotype I think we saw is with bladder pain. My friend and collaborator, Dr. Green, is a pain expert. So we have been troubleshooting the chronic UTI model where we can see a robust bladder pain phenotype. And the one we landed on so far is 3 repetitive infections using CFT 073. So in the wild type, you infect these mice 3 times over and over again, and it actually will trigger a long-term pain phenotype. You let them rest for 2 weeks, you measure abdominal pain using Von Frey 2 weeks later, and you are going to still see a persistent pain phenotype. And in the B2 knockout, this pain phenotype almost completely diminishes. So we're really excited for the potential to target this receptor as a pathway to treat UTI because we can simultaneously reduce bacterial load, inflammation, and also pain. We are currently investigating this possibility using a humanized mouse. I'm so sorry. Where we have an engineered mouse line to express the human version called MRGPRX2 in MRGPRB2 knockout and replace the mouse version with the human version. And you can see that the human receptor will rescue the phenotype back to the wild type level, which allows us to test drugs that are antagonists for this receptor. And we have so far tried one drug called Oslo. This is a natural product. And we have seen an effect on the 24-hour CFU. So this is pretty exciting. So I think I'm a little over time. My apologies for the cough. And we are overall looking at antimicrobial peptides as a signaling system. And I hope I have convinced you that the signaling function is just as important, if not even more important, as the direct antimicrobial function of these peptides. And among all the receptors that we've looked at, we have so far found that the mast cell receptor MRGPRB2 is actually performing a maladaptive function and triggering excessive bladder inflammation and pain during urinary tract infections. I'm out of time, but we actually do have several cool ongoing projects about around UTI in the lab. We are also looking at another type of innate immune cells called basophils, and we see a pretty cool phenotype there as well. Ridma and Zakia are looking at neuroimmune interaction in UTI pain and also bladder, the contribution of neurons to bladder immunity. I'm really excited about this. We are starting to look at the UPACs themselves and how they are responding to these host receptors using RNA sequencing. We have done this in the skin successfully, and we are doing that the same in the bladder in the future. I am personally very, very interested in the immunity and the functions of these peptides and cell types in the urethra, which is even more understudied than the bladder itself. And you all have heard the talk from my postdoctoral fellow, Dr. Maliha Batu, a couple weeks ago on her project in the role of defenses and mast cells in bacterial vaginosis. So I'm going to skip all my other slides and go to acknowledgement. So I want to thank the UTI team in our lab. In addition to the postdoc and PhD students, we actually have 15 undergrad researchers in the lab who have contributed substantially to the research program. I need to thank all my collaborators, my co-investigators, especially Dr. Denisko and the Denisko lab who taught us the UTI. Sorry, what is wrong with, what's wrong with my mouse? I have no idea. They taught us the UTI model, really held our hand and taught us, like all the standards of the field. We really, really appreciate it. And my collaborators and co-corresponding authors, Dr. Green and Pundir for the mast cell story. And here's my contact information, and I would like to take questions.
Thank you. Thank you. Awesome talk. We are running a little tight on time today, so I'm going to say that we don't have time for questions right now. But if you do have questions, please, Dr. Dong's email is on this slide. Go ahead and send her an email, and maybe y'all can have a great chat. We're going to move on to the second talk today. Dr. Kymora Scotland, who is Assistant Professor, Department of Urology at UCLA. Really looking forward to her talk as well. And I'll let you share your slides, Dr. Scotland, and we can get going.
Fantastic. Thank you so much. And thank you again for the invitation to chat today. I will just let everybody know that unfortunately I am in clinic today, and so I have a hard stop at 1 o'clock. And so I'm going to try to get through as much as I can, but happy to get any questions from everybody via email or any other way you want to contact me. Okay, so kidney Stones Harbor Bacterial Bunkers. These are my disclosures, and I'll go very quickly through this. We still don't know why kidney stones form. And that's important because they cause a lot of pain. The pathogenesis is really multifactorial. We know that there's some contribution of genetics and diet, even immunity. UTIs, as we just heard about from our previous speaker, and potentially bacteria. But really, bacteria in the sense that there is a small population of kidney stones called struvite stones, which we know are formed as a result of bacteria. Uh, Most stones, about 80% of them, are calcium oxalate stones. And those stones are not thought to be associated with bacteria, really. They're thought to form as a result of supersaturation. So basically, having extra crystals in the urine. Okay, and so that's our current picture of kidney stones. So you've got these crystals, they aggregate and they form stones, and they afflict about 10% of Americans every year. Now, I come from this as a urologist, thinking about UTIs and wondering if, in fact, there is a connection between kidney stones and the UTIs that we often do see in kidney stone patients. But typically, what happens, as you can see from this CT scan here, is that a patient will have a stone that is blocking or obstructing their ureter. And then they develop a UTI or develop sepsis. And the assumption is, well, that urine has been stagnant for some time. There's bacteria there, and that sort of formed a biofilm on the surface of the stone. And so that's why we sometimes see stone cultures that are positive. So as I mentioned before, typical infection stones are struvite stones or something called apatites. And they're formed by the action of urease producing bacteria. But when we see patients with calcium oxalate stones who have infections, those are often not the bacteria that we find. In fact, we find things like E. coli, Pseudomonas, which may not necessarily be urease producers, certainly things like corneobacteria, et cetera, et cetera. But what that led us to think is that potentially there's something else happening here. So are there data to support a role for bacteria in calcium oxalate kidney stone disease was the question that my lab started with. As I mentioned, several groups prior had already identified bacteria doing sequencing of calcium oxalate kidney stones. And that sort of reminded me, in the interest of time, I'll move through this. But that did remind me of something that we had done previously in my postdoctoral work, where we looked at the encrustation of stents. Now, stents are temporary tubes that we put in the kidney to drain the kidney before or after a kidney stone surgery, and they can become encrusted. And those encrustations look really lots like stones. We wanted to characterize them and identify bacteria within those encrustations. And so the hypothesis, became, well, maybe uropathogenic bacteria actually are playing a role here, and they might be promoting calcium oxalate stone growth, and that maybe they do this via the use of biofilm and biofilm components. So that was the first thing we wanted to do, was to try to identify bacteria within stones. And so we started working with a series of people, including a series of earth scientists and geologists, because it occurred to us that, kidney stones are like every other stone. And there are stones that exist within geologic circles known as stromatolites and travertines, which are stones that are formed by the action of bacteria. And so we started doing stratigraphic evaluations, looking at the layering in the stones, and started seeing things, if you can see my mouse here, within the organic layer of the stones that looked sort of biogenic. Those looked like cocci. And so that led us to, again, clarify our hypothesis that maybe what's going on here is you have this microbial mat activity. Microbial mat activity in geology is very similar to biofilm deposition everywhere else. And so we thought that maybe this was what was going on. And so the first thing we wanted to do was to get a better sense of what the substructure of kidney stones would be when we think about things like biomineralization. So, and that again is the deposition of minerals in specific patterns, and it's typically influenced by microorganisms, often bacteria. And so the first thing we did was to work with a company called Sigray, who had developed this imaging technique where they did basically 3D X-ray CTs and X-ray fluorescence microscopy, all, simultaneously to get a better sense of substructures. And we started to identify the mineral and organic substructures that we expect to see within stones, and started identifying what we called at the time pore structures that we thought might house bacteria. We'll put a pin in that. And so this is another look at the distinct sort of areas of nucleation. So what I want to show you here is down in the bottom right is a struvite stone. So again, struvite stones are known to be formed by bacteria. And you see these various nucleation sites here. You see these whorls where, individual biomineralization events were happening. But we start to see similar things happen. And these are all calcium oxalate stones. And we start to see similar things happening here. And then again, we see these, what we at the time were calling pore structures that we thought were, really potentially housing bacteria. Okay. We then moved to scanning electron microscopy to get a better sense of what was happening within the stones. Now, I mentioned previously that lots of folks had thought that, sure, sometimes stones can culture positive, and it's probably just biofilm on the surface of the stone. So in order to put, paid to that, we then fraction stones. So we obtained stones from patients. I'm a urologist. We fractured the stones, we fragmented them, and we showed that within the internal fragments of the stones, on scanning electron microscopy on SEM, we were able to see structures that again appeared biogenic, what looked like cocci and rods. And in order to have a better sense, we then shifted to ionic liquid coating SEM. Now, what happens with a lot of the SEM that is done is, you have to obviously prepare the sample, and that dehydrates a lot of live cells. And so by ionic coating, using stains that were able to identify bacteria, specifically cocci, as well as biofilm material, okay, that you see here, we were able to show that within the stones, we do see these, again, connecting sort of biofilm layers, as well as individual bacteria. These are bacteria nestled within what are called nanocrystals, which are known structures seen in biomineralization. What I want to just mention here is that of these 4 panels, okay, only panel B was culture positive. So what this means is that in patients, okay, from whom these stones were extracted, they had no histories of UTIs, but then the stones themselves, when cultured negative. So these bacteria and these biofilm layers were found even in the absence of any evidence of infection. Okay. And so we wanted to take a closer look at this and to try to determine whether, in fact, these were just bacteria that had somehow been incorporated into the stones during the process of biomineralization. So we did some live cell staining. And so we did two photon and regular confocal fluorescence microscopy. And what we're looking at here is the following. So on the bottom is a control whewellite sample. So calcium oxalate is also known as whewellite, and calcium oxalate exists within the world. It exists, not just in kidney stones. So these samples were obtained from a quarry. And you see here that, apart from their background fluorescence, you don't really see any staining. Now, what I'm showing here with the green staining is a stain for bacterial DNA, indicative of live bacteria. So here, this is just your wide field of a calcium oxalate stone. These are various fragments of the stone. And then we have 2 insets here. This first inset is actually here where we're looking at bacteria, and we're able to, in another slide I'll show you, see that bacteria is co-localized with mannose, which is typically seen in biofilm. And then here, what's I think really cool is that we not just identified bacteria within the structure, but you can see that it's really layering within the organic layers of the stone. So the purple here is the mineral sort of calcium oxalate layers, and then the organic layers, because kidney stones incorporate not just minerals, but also various organics. And so that is where the bacteria sort of lives. I mentioned here that we would look at mannose staining as well, and we were able to show that the cocci co-localized with all of this mannose staining. Okay, so we have bacteria within their biofilm communities. And then, we wanted to see what was going on within the stones. And so instead of doing just simple SEM, we did something called focused ion beam SEM or ion milling, which is basically a way of doing a three-dimensional look through stones. So we're cutting through the stones while using SEM. And here in blue is the mineral. In this sort of yellow-green color is the biofilm and bacteria. And you can see and appreciate that there are, again, these sort of voids, these holes within the structures. So we see those again, which we had seen previously in our micro CT. And this just shows a small movie of that where you really do see these voids within the structure. And then you see an individual bacteria embedded within a biofilm matrix. So that brought us back to work that was done several years ago now by actually a geologist, Bruce Falk. Bruce had come to looking at kidney stones from geology. I've since gone from medicine, looking at geologic stones. And what they were able to find is that those voids were actually areas of dissolution. So what we currently are thinking is that a lot of times what happens with calcium oxalate stones is that you have a mix of monohydrate and dihydrate stones. And calcium oxalate dihydrate stones can actually dissolve and then reform. And that in these areas of dissolution, we hypothesize, there's lots of activity and opportunity for bacteria to survive. Okay. And in work that we did with Henry Schreiber, who is also associated with the CAIRIBU group, we actually looked at kidney stones. Now, I want to put a plug for talking to people at meetings because I met Henry and talked to him at a CAIRIBU meeting, and he mentioned to me that he had been interested in kidney stones a while back and had extracted a bunch of bacteria from stones. And I thought, oh, how interesting. I'm interested in kidney stones. And so we've now we're just beginning a collaboration, but really we're able to use data that he already had to show that bacteria were able to be extracted from these kidney stones from patients with, again, no history of UTIs. And what we're able to show here is that, first of all, the bacteria within stones are often polymicrobial. So down in C, you can see that, per kidney stone, there are several stones where there are multiple different types of bacteria that they were able to extract. And then we see a lot of the usual suspects that we would expect to see in UTIs. Again, supporting this hypothesis that it's uropathogens and potentially specifically biofilm-forming uropathogens that may play a role here in kidney stone growth. So the question now is, well, how do these layers organize within the stones? Why would bacteria want to do this? And so we went back again to light field and Raman spectroscopy, which I'll talk a little bit about in a minute. So we started to look again at the layering of the stones, and we found something interesting. So what you see here are dark areas being organic and the lighter areas being mineral. And you already can start to see here that the mineral within these areas are they're fairly large crystals as compared to the regions, within the organic layer or very close to the organic layer, which are much smaller. So we saw that the domain sizes of the crystals formed right on top of that organic layer tended to be much smaller than those farther away from the organics. And so what does that really mean to us? So we started thinking that maybe bacteria is actually playing a role in nucleating crystals. So you have lots of small nuclei right around the area where the bacteria exist. And further away, you're able to have larger crystal sizes. We took a closer look here, and you certainly see, again, organic layers here, small domain sizes right around the organic layers. Further away from the organic layers, you're seeing much larger crystals. And this gives you a little bit of a better view just with, the bacterial layers removed. So then what we started doing was thinking about what that would mean for the stone. So this is not just a single bacteria sort of causing biomineralization, and then you have a piling on of minerals that, would happen as would happen with any other supersaturation event. What we're seeing is that bacteria seems to be really throughout the stone causing different nucleation events, extending the biomineralization. And so now we're starting to see these, as I showed you earlier, these separate nuclei within the stones. In order to sort of look at this at the same time, both with CT and with Raman, we had to develop some methodology. And so a lot of the work that we're doing now is trying to develop a real-time Raman spectroscopy that we're able to correlate with micro CT. So this is just to show you a little bit of what we're doing now, and this publication will hopefully be out this year. So looking at the CT slices. So this is a slice of a calcium oxalate stone, and what we're able to show is that moving from layer to layer, we have bacteria and we have minerals. It's not just within the organic layer, as I showed you, but even within the mineral layers, there's a little bit of biofilm. There's a little bit of biofilm throughout this structure. And so we think that what happens as the stones continue to grow and the layers continue to form is that you have biofilm structures that are at least triggering an increased growth rate of these mineral layers. Okay. And then, as I mentioned, we're trying to do real-time Raman. So we have some correlative work that's already showing where within the organic layer, certainly you have bacteria, but even within the crystalline layers, you have at least some biomass that is suggestive of biofilm materials. Okay, and so what we're seeing at this point is that bacteria are located within and not just on stones. They survive within these calcium oxalate stones. And so the next step for us is to try to figure out if biofilm is actually necessary for that phenomenon. I'm going to really run through the rest of this data just to show that we're doing some work in vitro, trying to identify what it is that bacteria is doing at a crystallite level. And we were able to show that bacteria do in fact localize to crystals. We were able to show that bacteria causes an increase in the formation kinetics. So you've got an increase in the size, the number, and the aggregation of these crystals in vitro. And then I'm going to scroll through this really quickly. We do see that these are, in fact, in the presence of bacteria, the same crystals. We've confirmed this with Raman, we've confirmed this with EDX. But what's interesting about what we see is that we also see these voids within crystals. Again, a different phenomenon, obviously, but we see these, and this is again FIBSEM, where we see these areas of crystalline material with a central, what we're calling etching. So these are crystals grown in the presence of E. coli and Pseudomonas, and we see this central dropping out, this hollowness within the crystals, and we think that what that really correlates to is the organic material that is helping to twin these crystals and is helping them to aggregate in a fashion that is much more secure than what we would see normally. So I'm just going to end here by showing you what happens with regular aggregation with just control crystals. The crystals sort of stack side by side. In the presence of bacteria, we start to see this twinning where the crystals really grow into each other, making for a much stronger crystal, and they sort of, they really aggregate together. And so we'll end here by saying that we think, this also happens in vitro. We've developed a model, a microfluidic kidney model, where we're looking at all of this. And hopefully we'll have some data to show you later on this year. I will stop here just because of time, but the questions that we're trying to really answer right now is, of course, why bacteria do this. We have some thoughts, what that means for the cell, and how that helps with our understanding of biomineralization, not just in kidney stones, but in other organs. I'll leave here with my acknowledgements page, because obviously this work is not done just by my group, and I'll stop here.